Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers, everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. We're thrilled by the overwhelming support for our episodes featuring top-notch experts delving into various biological realms. Thanks to the feedback from higher biology students, we're launching shorter, punchy revision episodes. Get ready for quick summaries of key areas to turbocharge your exam prep. These short episodes can support your exam preparation and are not intended to replace other important aspects of your revision. Microorganisms, including archaea, bacteria, and some eukaryotes like yeast, are powerhouses of metabolic activity. They're used in biotechnology because of their adaptability, rapid growth, and the ease with which they can be cultured. But to harness their potential, we need to understand the conditions they need to thrive. In this episode, we'll explore the nutritional requirements of microbes, the importance of sterile conditions in fermenters, and how factors like temperature, pH, and oxygen levels are carefully controlled. You'll also learn how microbial growth is measured and what happens during each phase of their population growth curve, from lag to exponential, stationary, and finally death. So whether you're brushing up before the final exam or consolidating your understanding, stay tuned as we break down everything you need to know in the environmental control of metabolism key area. Microorganisms use a wide variety of substrates for metabolism and produce a range of products from their metabolic pathways. We use microorganisms because of their adaptability, the ease of cultivation, and the speed with which they grow. Now, the growth media that microorganisms are cultured or grown in must contain raw materials for biosynthesis as well as an energy source. Most microorganisms are able to produce all the complex molecules they require for biosynthesis, such as amino acids, vitamins, and fatty acids. But other microbes need these to be supplied to their growth medium. Another complex compound which may be added to the growth medium is beef extract. Now that might sound a bit strange, but beef extract is added to the growth medium because it's a really rich source of complex organic compounds that some microorganisms can't synthesize on their own. Specifically, tends to provide amino acids, peptides, vitamins and minerals, and those nutrients support the biosynthesis of essential cellular components, especially for microbes that have more demanding nutritional requirements. It's commonly used in general purpose media like nutrient broth or agar that you might have used in the school lab. When we talk about energy sources, they're either going to be chemical from food or respiratory substrates, or light for photosynthetic microorganisms such as algae. The conditions in which microbes are cultured must be monitored and regulated closely. Large-scale production of microbes is done in a fermenter. Now, we can think of a fermenter really as a large sterile container or bioreactor used to grow microorganisms under controlled conditions to produce useful products. And it's used in a range of industries to produce these products, from the pharmaceutical industry producing antibiotics, the food and drink industry for brewing beer and making yogurt, right through to the biofuels industry to produce ethanol and biogas from microbial fermentation. These fermenters are designed to create the ideal environment for microbial growth and metabolism by carefully monitoring and controlling temperature, pH, oxygen levels, nutrient supply, and sterility. Now, we need to control the temperature for optimum enzyme activity. The pH is kept stable using buffers, acids or alkalis. The oxygen levels are maintained by aeration to ensure aerobic microbes are able to aerobically respire. And nutrient supplies are controlled to ensure enough raw materials and energy sources are available. And it's important that the inside of the fermenter is able to be kept sterile to prevent contamination and competition from unwanted microbes that might risk spoiling the overall product. These fermenters and the ability to control these different elements allow microorganisms to be grown quickly and efficiently, making them ideal for large-scale industrial production. Growth of a population of microorganisms occurs when the rate of synthesis of organic matter is greater than the rate of breakdown of organic matter. Growth is an irreversible increase in dry biomass. Now, increase in dry biomass is a more reliable indicator of growth than gain in fresh biomass, because an organism's fresh biomass varies depending on water availability. The generation time, or the doubling time, is the time that it takes for a cell or a microbe to divide into two cells. And a microbe's pattern of growth varies over time, and there's 4 distinct phases of growth that we learn about. The first phase is the lag phase, where enzymes are induced to metabolize new respiratory substrates. Cells adjust to growth medium and show an increase in metabolic activity. There's really little or no increase in the cell number at this point during the lag phase. That starts when we get to the second phase, the log or the exponential growth phase, where cells multiply and grow at maximum rate. The population doubles with each cell division. This phase has got the most rapid growth of microorganisms since there's a plentiful supply of nutrients. Then when we get to the stationary phase, it's a phase that secondary metabolites are produced, such as antibiotics that can be produced by fungal cells. And those secondary metabolites provide microbes with an ecological advantage by allowing them to outcompete other microbes. But during this phase, the stationary phase, the rate of production of new cells is equal to the death rate of old cells, and the population remains steady as nutrients begin to run out. Then we hit the death phase. During this phase, there's a lack of nutrients and a real build-up of toxic metabolites. Cells die at a constant rate, and the number of cells dying is greater than the number of new cells produced. All sounds a bit grim. Now let's talk about how microbial populations are actually measured. Well, viable cell count involves counting only the living microorganisms, while total cell count includes both viable and dead cells. This distinction is important because only viable cell counts show a death phase, where the number of living cells decreases. Microbiologists or lab technicians might carry out these counts in industrial settings, like pharmaceutical or food production, to monitor the health and productivity of microbial cultures. And it's crucial for ensuring product quality, safety and consistency during fermentation or other bioprocesses. And once we've counted those cells, how do we actually make sense of the data? Well, microbial cell numbers can be plotted in different types of graphs, including a normal arithmetic scale and a semi-logarithmic scale. What's useful about a semi-log scale is that it shows the log or exponential growth phase as a straight line, making it easier to spot trends and calculate growth rates. This method is especially handy in both industrial and academic settings, where understanding microbial population dynamics is key to successful monitoring and control. So, we've covered how environmental conditions are controlled to optimise microbial growth, the stage of the microbial growth curve, and why microorganisms are so valuable in industrial processes. If you found this helpful, be sure to listen to some of our other episodes where we tackle other key areas of the higher biology course. Until then, keep revising. So hit subscribe on whatever platform you get your podcasts. so you don't miss the rest of our short summary episodes nor our regular episodes with leading experts. And remember, this quick summary is intended as just one element of your broader revision plan. There are lots of resources available via Glow such as Scholar or WestOS, as well as BBC Bitesize. Or why not check out one of Lecky's great revision books and use the code LeckyPodcast40 to get 40% off on their website www.leckyscotland.co.uk.